Okay, Dr. Gordon, she's professor in the Department of Physiology and Biophysics at the University of Washington School of Medicine. She earned her PhD at Brown University in 1993 under Anita Zimmerman, and then did a postdoc as an HHMI senior fellow with Bill Zagoda at the University of Washington from 93 to 97, where she studied the structure and function of cyclic nucleotide-gated channels involved in sensory transduction. Excuse me. In 1998, she accepted an assistant professorship at the University of Texas Southwestern Medical Center. But in 1999, she moved back to the University of Washington School of Medicine, where she was an assistant professor until 2005, when she was promoted to associate professor, and then finally to full professor in 2013. Dr. Gordon's research is focused on uncovering mechanisms by which sensory signals are converted into messages that can be understood by the brain by looking at how signal transduction cascades provide the energy to control ion channel function. She has won too many awards to list, but in fact, she has an award named after her from the Society for General Physiologists called the Sharona Gordon Award for Transformational Leadership. She has been and is currently well funded by the NIH with an R1 wrapping up now and an R35 underway, and of course is published prolifically using a variety of techniques, including genetic code expansion methodologies. Importantly, she is an internationally recognized leader for her efforts to promote equity and inclusivity in the science community, particularly among aspiring women scientists and students. So thank you, Dr. Gordon, for joining us today. We are excited to have you. Please take it away.
Thank you. Can you hear me?
Yep.
Okay, great. So it's 7:30 in the morning here in Seattle, and I'm both really excited to be following James, who's been a real role model in the field and personally for me an amazing collaborator, and a bit anxious that my children and dogs are going to be in the background and trying to be disruptive. So I apologize in advance. These are not the droids you're looking for, just ignore them. So I couldn't also have asked for a better introduction. Today I'll be telling you about some of the applications, or one of the applications that we've been using for ACD, and a little bit for ANAP, and how we use them to try to understand the energetics of allostery, of how conformational changes are promoted by ligand binding in proteins. So I want to take you back to the setting of why we would bother to do these actually pretty difficult experiments. And so here I'm showing you a figure from the Horgan and Aldrich paper from 2002, in which they're trying to describe the gating of the BK, or large conductance calcium-activated potassium channels. And they've condensed the Monod-Wyman-Changeau kind of gating scheme, the kind of scheme that was used in the 1950s to describe the binding of oxygen to hemoglobin. And they've adapted it for ion channels and put it in a form that really makes structural sense. And that is that they've described these calcium-activated potassium channels in structural terms as having a pore domain depicted here. And the pore can be either in the closed configuration where the gate is closed, or the open configuration where the gate is open and now allows the flux of ions down their electrochemical gradients. And that equilibrium in the pore domain is described by some equilibrium constant L. They also have a voltage sensor. These are voltage gated channels as well. And the voltage sensor is in equilibrium between its resting state and its active state. And this equilibrium at hyperpolarized potentials favors the resting state, and at depolarized potentials favors the active state. And this voltage sensor with its own equilibrium is also coupled to the pore gate. And there's a third structural element in these channels, which is the calcium binding site. It's an intracellular calcium binding ring, and it's in equilibrium between its apo or resting state, or its bound and hollow state. And its equilibrium has its own equilibrium constant, and that calcium binding domain also is coupled to the pore and influences the pore gate, and perhaps is also coupled directly to the voltage sensor. And so this is a nice model because it allows you to see in structural terms what we're measuring in functional terms. It's also a great model because it shows you how the measurements that can be made experimentally can be used to understand gating. And that is that we have patch clamp analysis, electrophysiology, that lets you, with great temporal precision, with sub-millisecond precision, measure the equilibrium between the closed and the open states of the pore. We also have the ability in channels that where the ion conducting pore has been compromised, or in which experiments in which no conductive ions have been included, to measure the gating current. The voltage sensor is itself charged, so as it moves through the membrane in response to depolarization or hyperpolarization, the charged amino acids moving is a current, and you can measure the gating current that allows you, again, to look at the distribution of those voltage sensors among the resting state and the active state. And because we can measure the conformational equilibria of both this ionic gate domain and the voltage sensor domain, you can infer what's going on in the calcium binding domain here. And in this way, Horgan, Aldrich, and others like Dan Cox have been able to build really beautiful models trying to understand how these calcium-activated potassium channels work. Now, in my lab, we've been interested for a long time in a related channel called trip channels, and there are about 30 different trip channels. Very few of them are gated by voltage. Some of them are gated by calcium, but they're also gated by a host of other compounds, and I'll briefly tell you about one of them. So this is a representation of the TRPV1 ion channel that's gated by noxious stimuli, so it's a pain receptor in your peripheral nervous system. Like the calcium-activated potassium channels, it has a pore that can be in a closed or an open position, and it has a region with homology to the voltage sensing domain. In the case of TRPV1 channels, it's not gated by voltage. Instead, it binds capsaicin, which is the pungent extract from hot chili peppers. And so when it binds this capsaicin molecule, the capsaicin biases the equilibrium of the voltage sensing homology domain between the resting and the active state. Now, it also has all of these other domains, some of which are likely to bind, or we know to bind toxins like insect toxins and spider toxins. And these toxins bind to the outer pore turret and activate the channels. It also has regions like an intracellular anchor and repeat domain, which contain reactive cysteines. These reactive cysteines react with pungent extracts from mustard and garlic, which then modify the cysteines and activate the channels. It also has other intracellular regions. I don't want to go through all of the regions, but what you can see is that there's a lot of them, and that they probably mostly couple to the pore gate to give energy to drive the opening conformational change in the pore gate. And perhaps they're also coupled to each other. And the point that I'm trying to make looking at this is that we can measure directly the conformational changes in the pore using electrophysiology, using patch clamp, but we don't have any methods that really let us measure the conformational equilibria in any of these other sensing domains. In particular, there's a lot of interest in understanding how these channels are gated by heat. I probably don't have to convince you, you've experienced that spicy chili peppers feel hot. The reason they feel hot is because your brain doesn't know whether the channels were activated by heat or whether they were activated by hot chili peppers. It's activated by both, and yet we know almost nothing about the mechanism that heat uses to activate these channels, whether it even acts on any distinct structural domain, or whether it's a more distributed effect across the entire protein. And so there are a lot of mysteries to how these channels work. And with standard patch clamp electrophysiology, we just don't have a tool that can tell us what's going on in any of the individual sensing domains, let alone at how they might be coupled to the pore or how they're coupled to each other. And so our long-term goal, myself working with Bill Zagoda's lab, has really been to understand how activation or the equilibrium in any sensing domain or in any group of sensing domains impinge energetically on the pore, allowing the pore to do a calculation to determine what its open probability should be in response to all these different equilibria in the different sensing domains. And so that can be illustrated simply with this diagram. And this is based on a diagram originally made by Stefan Stoll, who's a DEER or EPR spectroscopist at the University of Washington. And it depicts this clamshell-shaped protein in equilibrium between a resting state and an active state. And that equilibrium can be biased by the presence of a ligand. And so in this case, the resting state would be mostly in the absence of ligand, and the active state would be mostly in the presence of ligand. And in our case, as James described really well, we can measure the presence in the resting state by using a pair of FRET probes. So a donor and acceptor at the top of the clamshell. And in the resting state, they're far apart. And so we might have very little FRET. And in the active state, the ligand might bind here in the clamshell's mouth. You'd get a conformational change that brings these two probes close together, and we can measure the distance between the probes, and now it's shorter. So what you might see if you looked at the probability of being in any given state as a function of distance. So the probability in the resting state at being in the short distance in the closed clamshell conformation might be pretty small, but it's a Boltzmann distribution. There'd be some probability of being in that closed clamshell state, but mostly the probability would be the area under this curve of being in the long distance state that reflects the open clamshell. And then when you had binding of ligand, you might get a shift of the probability from the open clamshell to the closed clamshell. And so you might measure something that looks like this, where now instead of being mostly in the closed clamshell in the presence of ligand, mostly in the open clamshell in the presence of ligand, you've shifted to the closed clamshell. And so these are exactly the kind of probability densities that we can measure for the pore domain using electrophysiology. And our goal has been to develop methods in which you can do these same kinds of measurements in any domain that you want to, even electrically silent domains. And so to do that, we use FRET. And again, James has already introduced transition metal FRET, but the idea is that this is an intracellular measurement where we have a donor probe in one position and an acceptor probe in another position. And as he told you, the acceptors that we're using are typically transition metals. And so why are we doing that, and how are we doing that? So the how that we're doing that is either by engineering in a dihistidine motif on an alpha helix at I and I +4, or on a beta sheet, a beta strand at I and I +2, or by using this TTAC, which is a cysteine reactive cyclin ring. And so we can react it with an engineered cysteine at any position on our protein, and that gives us the transition metal at our position of interest. So that's the how. The why is really demonstrated over here. This is a plot of the Forster equation, which shows the FRET efficiency as a function of distance. So at very short distances where the donor and acceptor are close together, you have 100% FRET, which would mean that this acceptor transition metal 100% quenches the donor. So unlike FRET with two visible light fluorophores, when you have one visible light donor and a transition metal acceptor, FRET appears only as a quenching of the donor. So when you have 100% FRET, you would have no fluorescence from the donor. At very far distances, you have no FRET, and so you'd see the same fluorescence emission from the donor that you would see in the absence of any acceptor. And in this regime over here, you have a steeply dependent dependence of the FRET efficiency on the distance. The steepness here depends on the 6th power of the distance. And the place where it's most sensitive, the sweet spot for FRET, is the distance at which you get 50% FRET efficiency. So you can see here it's really steep, but if we were looking at distances over here or over here, it's not steeply distance dependent. And so this 50% FRET efficiency regime has a name. It's called the Forster radius, or the R0. And the key for transition metal FRET is that this R0 is in the 10 to 15 angstrom range. Unlike classical FRET with 2 visible light fluorophores, where this R0 would be way out here, even beyond my slide, in the 50 to 70 angstrom range. So our ion channels are 50 to 70 angstroms on a side, and FRET probes that let you see only distances at 50 to 70 angstroms would require us to put our probes at the extreme ends of the channels, and those don't let us see conformational rearrangements within just one domain. And so a huge advantage of transition metal FRET is that it lets us look at these conformational rearrangements in small sensing domains within our proteins. It has a lot of other advantages as well. One of them is that the transition metals have multiple transition dipoles, making it pretty much immune to the orientation problems that standard FRET has. So we can do this. This is just another diagram of how we covalently link this TTAC, this cycling ring, to our protein. This is the leaving group. The cycling ring gets attached to our protein, and it'll bind a transition metal. So this also, James covered how we introduce these transition metals using genetic code expansion. All of our plasmids that make the synthetase have been developed by others. Again, for ANAP, it's Abhishek Chatterjee when he was with Peter Schultz. And for ACD, it's James in collaboration with Ryan and the Center for Genetic Code Expansion. So here's the protein that we're looking at, maltose binding protein. And we typically focus on 2 different FRET pairs. One at the top of the clamshell, and in the apo, or in the absence of ligand, it's in the open clamshell state, and the distance is about 21 angstroms. In the presence of ligand, it's maltose bound right here. It decreases about 17 angstroms to 13 angstroms. We also have another pair of FRET probes that we can put down at the bottom. In this case, in the apo state, or in the absence of ligand, it's a short distance, and it gets longer in the presence of ligand, about 3 to 4 angstroms. And so let me show you what our typical data look like. So here we have our open clamshell and our closed clamshell in the presence of ligand. This FRET pair, here's the donor, here's the acceptor. The fuzzy clouds are just predicted distribution of the rotameric distribution of the donor and the acceptor. You can see the clouds are actually pretty big relative to the size of the proteins. And so in this case, we're using ACD. We measure the fluorescence in a cuvette. We add, in this case, copper TTAC, and we see quenching that is this 20% quenching means the FRET efficiency is 20. And then we can recover it by removing the transition metal acceptor using a reducing agent. When we do this for ACD, it looks like this. When we do this with ANAP, it looks like this. They're quite similar. This is the top of the clamshell. At the bottom of the clamshell, instead of getting closer together, the donor acceptor pair get farther apart. And so we see less FRET in the presence of ligand, which are the closed symbols. So what I told you is that we're not really interested so much in the distances. We have crystal structures. We know the distances. What we're interested in are the energetics, the distance distributions. So we don't need to know the distances in the resting state and the distances in the active state. What we need to know is this information here. What's the distance distribution in each of the 2 states? So this tells us the equilibrium constant between the resting and active states in the absence of ligand. Here is the resting state between the resting and active in the presence of ligand. And the difference between these 2 is the delta delta G. the difference in energy, the extra energy contributed to activation by ligand. So we don't use anymore the steady state FRET that I just showed you. We use fluorescence lifetime measurements, also introduced by James. And conceptually, what this means is that when we excite in delta function with a pulse of laser shown in blue, we can measure the emission as a probability over time. So most of the light is released here, and then less and less and less light. And this has a characteristic exponential decay, which James told you for ACD is about 15 or 16 nanoseconds. Now, when we introduce an acceptor, it's another pathway for that excited state fluorophore to lose its light. And so the lifetime in the presence of the FRET acceptor gets much shorter. And so this is what you might get, a single exponential, if you had only one lifetime. So if you had only one distance, you might get something that looks like this. But we've told you that you don't have just one distance. You have a distance represented by the resting state and a second distance represented by the active state. And so what you actually get is something like that that's shown here. And you might not be able to tell by eye on this linear plot, but this is the sum of two exponentials. And you can see that more clearly if I plot it on a semi-log plot. Now you can see that there's one exponential with less with more FRET shown by representing the closed state, and a longer exponential representing the open state. And so what we want to be able to do is get the amplitude of these two exponentials, which tells you the amplitude or the representation and probability among the two states. So we don't actually use fluorescence lifetimes in the time domain. We measure them in the frequency domain, which is more complicated to explain, but we get really exactly the same kind of data. So the data look strange. What we get is something called a Weber plot, in which we're not just measuring the lifetimes in response to one frequency, we're measuring at lots of different frequencies. And what we get is a phase delay and a modulation ratio. And the analogy that I can make to make this sound not so alien is a lot of you probably know about X-ray crystallography. And in X-ray crystallography, the data also can be represented by phase and modulation, and that the lunes on the X-ray graphs that you see of all the different intensities represent the modulation, and the problem that they usually have is getting the phase. And so from these experiments, we get both a phase and a modulation, and we can plot those as a function of frequency. For ANAP, they're shown here. The data themselves are the symbols. And if we tried to fit the ANAP data as a single exponential, we'd get these solid curves and it wouldn't fit. What you need are multiple exponentials. Two exponentials give you the dash curves. And what this tells us is that ANAP is not a single exponential decay fluorophore. And so in addition to having these really short lifetimes, which reduce our dynamic range, the multi-exponential lifetime really complicates our analysis. ACD is like a godsend. So when James first told us about ACD, because we don't read the literature and we didn't know about it from reading, he told us, and it's amazing. It's like the miracle fluorophore. So here we're showing the phase delay as a function of frequency and the modulation ratio as a function of frequency. The fit with a single exponential is the solid line, and it fits beautifully. So not only is it beautifully single exponential, but it also has this 16 nanosecond lifetime. Remember that FRET shortens the lifetime. So if you're starting out with a 1 or a 3 nanosecond lifetime, and you want to measure something that has a lot of FRET that, say, decreases the lifetime 80%, it would be really difficult. But when you're starting out with a really long lifetime, it really increases our ability to measure a lot of FRET. So this is what the lifetimes look like in solution. What do they look like in our proteins? So here we have a maltose binding protein that I showed you before, and its fluorescent label ACD is at the top of the clamshell. So that has some fluorescence lifetime even in the absence of acceptor. and it's represented by the data shown here. It's fit with a single exponential decay. It has a 16-second lifetime. So the next thing that we do is we interact this with copper on TTAC, so it reacts with a single cysteine that's present in maltose-binding proteins. I'll say briefly that it wouldn't matter if we had other cysteines, because the R0, the distance over which this works, is so short that even if we had other transition metals at other places in the protein, they'd be essentially invisible for the purposes of our experiment. So we put in the acceptor and we don't have any ligand around, and the shortening of the lifetime is seen as the right shift of the curves. So in the presence of the acceptor, the data are these bow ties. And then we can add maltose, and you see a further shift. This is on the top of the clamshell, so we have more FRET as the donor and acceptor move closer together. What's really nice about TTAC is that you can, it's cysteine reactive, it's a thiol, so you can reverse it with a reducing agent. We put on TSEP and we get these open symbols that perfectly overlap with the original donor-only data, so we haven't just damaged or hurt our fluorophore. So these are what the data look like when you get more FRET with ligand. With the donor-acceptor pair at the bottom of the clamshell, you get less FRET with ligand. And so here in the APO or ligand-free state, you get a lot of FRET. When you put on maltose, you get less FRET. So I want to stop for a minute to talk about the fits to these data, these smooth curves. So, oops. Yeah, so the donor-only are really well fit by a single exponential. But in this case, I'm showing you that a single exponential doesn't fit the donor and acceptor very well. So why is that? So this fit is with a model. And what I haven't told you is that the model assumes a Gaussian distribution of the distances between donor and acceptor. And so here is the probability density as a function of distance. And what a single exponential requires is that you have a perfectly collapsed distance distribution with zero standard deviation. So zero standard deviation means that every donor and acceptor are at exactly the same distance. And this is not a physically realistic model, because even if it were just the thermal wiggling of the rotamers or the thermal wiggling of the background, you would expect some distribution around a mean for the distance between donor and acceptor pairs. And sure enough, because a single exponential doesn't fit, we can fit this with a model in which we do have a Gaussian distribution that has a finite standard deviation. And when we fit with a model that has a finite standard deviation, the fits of our of the model to the data give the dashed curves. And now, for the first time, tell us something about heterogeneity at this conformation, where we have a sigma and an average. So we have the center and we have the standard deviation. And so we get this for each individual state. So we can do this without any ligand and get this heterogeneity for the apo state, or for in the absence of ligand, and we can do it again in the presence of ligand. And in the interest of time, I'm going to go pretty fast. Here are fits to the data that we collect for the top of the clamshell, in which each individual experiment is shown as a thin string, and the average among the experiments is shown as the thick string for the absence of ligand and for the presence of ligand. Now, maltose-binding protein is a really weird protein, which in the absence of ligand really is almost completely in the open clamshell, and the presence of maltose almost completely in the closed clamshell. So that lets us see very well what happens when you have entirely one state or entirely the other state. But what we really want to be able to see is what happens when you have a protein in which you don't have entirely one state or entirely the other state, but when in either ligand-free or the presence of ligand, you have a distribution between the two states. Can transition metal FRET do that for us? So one of the limitations is really demonstrated here. So our donor is shown in gold, and our acceptor with its expected rotameric distribution is shown here, with the presence of the metal ion itself shown as the purple dots. this is a big cloud. And the big cloud for both the donor and the acceptor give us this really pretty broad standard deviation. The sigma is in the neighborhood of 6 angstroms. So a 6 angstrom standard deviation means that for the donor acceptor pair at the bottom of the clamshell, which are separated, the beta carbon to beta carbon distances in the crystal structure are separated about 3 to 4 angstroms, we can see the difference, but it's hard. So what we really need is a way to make these distance distributions more narrow. And so to do that, we go back in time, in fact, to a previous approach, which is to bind our metal to a dihistidine motif. And so here we have the exact same donor with the same donor cloud, but now our dihistidines are essentially a bivalent attachment to the transition metal, and our rotameric distribution that we expect is much smaller. So can we use this to narrow the distance distributions and see what it would look like if we didn't have everything in just one state or everything in the other state? And so the answer is yes, we can. And so here, again, we're using the exact same donor of ACD. This is now the donor acceptor pair at the top of the clamshell. And our acceptor is a copper that'll be attached to the pair of histidines. So these are the data in the absence of any metal, so donor only. Now I add copper. So it's a small shift. The R0 with copper isn't as large. And so we get less quenching. But now what we get is a distance distribution. So the fits are shown to this distance distribution. And its sigma is now only about 1.5 to 2 angstroms. So this is what it looks like in the absence of maltose. Now we add a saturating concentration of maltose. The data are the points, and the fits to this magenta distribution are the solid lines. And what you can see is that now we can actually see that the distance distribution is narrower in the presence of ligand than it was in the absence of ligand. Okay, but does this let us see what happens if we have a mixture between the two states?
Hey, Sharona, we're about 5 minutes over at this point, so if you could wrap up.
Okay, I'm wrapping up, and we did start 5 minutes late. Okay,
cool.
So
I'm sorry, thank you.
That's okay. So we can see if we add a subsaturating concentration of maltose just below the EC50 or just above the EC50, that we can indeed measure the distribution where we see some of the probability in the open clamshell and some in the closed clamshell. And so we can also do this using a different approach, using a phasor plot, which does not need to assume any particular shape to the distance distribution. So in conclusion, the transition metal FRET, using a time-resolved approach, using lifetimes, lets us see the average distance, as well as the heterogeneity between for any given state, as well as the distribution among states. The model-free phasor representation agrees with our Gaussian assumptions. And what we've learned is that we have more heterogeneity in the absence of maltose than in the presence of maltose. So again, to thank the people, all of this is a collaboration with Bill Zagoda and with James Peterson and Ryan Mell and the folks at GCE for All, and also thanks to the funders. And it sounds like there might not be time for questions, but I would be happy to talk to anybody about this afterwards.